Welcome to today's webinar on-farm anaerobic soil disinfestation for the management of soil-borne pathogens in high tunnel production systems. I'm Alice Formiga of eOrganic, and this webinar will last about 45 minutes, and then we'll have another 30 minutes for questions. Today, I am very pleased to welcome two members of a research team who have been investigating how to optimize anaerobic soil disinfestation as part of a NIFA OREI-funded project. Francesco Di Gioia, who is an associate professor of vegetable crops science focusing on the development of sustainable vegetable production systems, and Caterina Roman, who is a master's student finishing her degree in plant pathology. They are both from the Pennsylvania State University. So with that, I'm going to turn off my video and hand over the presentation to Francesco.
Thank you, Alice, for the introduction, and welcome everyone. Thanks for connecting to this webinar. I just wanted to take a couple of minutes to mention that this is part of a webinar series, and we have already done a few webinars that you can find on the eOrganic platform. And I also want to mention the fact that this work is funded through a project that it's like the funding agency is the OREI program, the Organic Research and Extension Initiative. And you see the award year, the number of the award year. And this is really a team effort, a multidisciplinary project in which we are working between the US Northeast and the US Southeast with vegetable and strawberry cropping system, of course, everything organic. And we have people that have different expertise that are working on different questions, different aspects of the project. For the work that we are presenting today, I wanted to really say thank you to a few people that contributed to the work that we are presenting, and in particular, Kati Demchek, who is a small fruit specialist and senior extension educator here at Penn State, Leah Frank, who is a Penn State extension educator, Joe Honorafel, who is a PhD candidate, co-advised between my lab and the lab of Dr. Francisco Dini Andreotti. And then Dr. Mia Cantor, who is a nematologist here at Penn State, is not part of the project, but has been helping us with counting nematodes and assessing the impact of ASD on nematodes. So we are really grateful for their support, for their help. For the presentation today, the goal of this presentation is really to provide some basic information on emerging soil-borne pest and pathogen issues that are affecting specialty crops in high tunnel production systems, in particular here in Pennsylvania, but we could say that the same issues are present within the entire Mid-Atlantic region, US region, and also in other places within high tunnels. And the other aspect that we want to show today is really how we implement ASD and how effective it is ASD in managing some of those soil-borne pests and pathogens. So we will have Caterina will start soon with an introduction on the emerging issues, soil-borne pest and pathogen issues. And then I will talk about how we implemented ASD on different farms using both clear and black plastic. And at the end, Caterina is going to present the results and show the efficacy of ASD in suppressing specific issues in those on-farm studies that we have done. So with this, I'm gonna end the control to Caterina. And Caterina, you can go ahead.
Okay, hi everybody. As many of you know, our high tunnels offer a lot of benefits to growers in our season extension, as well as increased fruit yield and quality. However, high tunnel production does have some constraints. While these structures were originally designed to be movable for the rotation of land that they are placed on, most growers keep them stationary at the moment and intensively cultivate our crops underneath these tunnels. This intensive cultivation, as well as a lack of rainfall and fertigation, can create issues in soil fertility, namely the accumulation of salts within the soil, which can stress our plants if levels become high. Additionally, and most notably of concern, is the increase of soil-borne pathogen pressures. This is mainly due to the monocropping of high-value crops such as tomatoes, peppers, and strawberries, as well as a lack of freezing temperatures within the high tunnels, since these structures tend to modulate the climate and raise temperatures, not allowing for the freezing temperatures that would die back some of the pathogen populations we're seeing. Additionally, there is limited management strategies that we can use within these tunnels. Growers don't tend to cover crop and crop rotate as much as we see in the field, as well as there being additional restrictions for the pesticides that we can use within high tunnel covered structures, and equipment not being able to readily fit in structures. And to kind of get an idea throughout Pennsylvania what some of these issues might be, the Gugino lab conducted a high tunnel soil survey of 90 farms throughout 17 of our counties from 2019 to 2021. And this was a targeted survey of tunnels with known or suspected issues as part of a larger study that we conducted with Ohio State that was recently published. This study aimed to evaluate the detection of Colotochum cucoides, verticillium wilt, verticillium dahliae, Pseudopranicida lycospiraceae, and terrestris, as well as meloidigine species. And to give you an idea about what some of these pathogens cause disease-wise, it is important to note that among soil-borne pathogens, the above-ground symptoms are quite vague in appearance, causing plant wilting, stunting, reduced yields, and sometimes chlorosis of the leaves. But it's not until we pull up the root system of the plants that we start to see more characteristic symptoms of these pathogens. On the left here is a picture of corky root rot symptoms caused by Pseudopranicida lycospiraceae and terrestris. This causes very characteristic corky bark appearance of the taproots, as well as a lack of fibrous roots and rotting of the fibrous roots. It's really the lack of the fibrous roots that creates the symptoms that we're seeing, as this limits water and nutrient acquisition of the plants. On the right side here, we see black dot root rot, which is caused by Colotochum cucoides, which is also one of the causal agents of anthracnose fruit rot. However, for the duration of this talk, when I discuss Colotochum cucoides, I'm referring to it in its soil dwelling stage, which causes lesioning as well as reduced fibrous roots of the root systems of tomatoes as well as potatoes. And sometimes you can even see the pathogen itself creating these black microsclerotia structures. Additionally, we have root knot nematodes, which are considered obligate parasites, requiring a living host to survive and reproduce. These pathogens can be quite tricky to treat, since the majority of the life stages of this pathogen are within the root system. And when we try to address this pathogen, we're mainly looking at managing its soil dwelling stages of the eggs and the J2 stage. Once that J2 enters the roots, it's very hard to curatively treat, and it will insist and cause that characteristic root galling that we tend to see with root knot nematodes. Getting back to our soil survey results, 88 out of the 90 high tunnels that we looked at least one pathogen. Again, this was a targeted study of suspected or known high tunnels with issues, but this really highlights how pervasive some of these diseases can be. In addition, I want to direct your focus to this Venn diagram, where 19 of the tunnels that we tested had all 4 pathogens, and another 24 had 3 of the pathogens. The results of this multiple pathogens occurring at once mirrored what we saw within Ohio, where 89.8% of all the high tunnels tested had 2 or more pathogens present at once. And really, this leads us with the major takeaway that high tunnels, that having a management strategy that is effective for more than one pathogen within high tunnel tomato production, as well as other high tunnel production, would be highly beneficial for growers to manage the complex issues they're facing. Additionally, focusing on plant parasitic nematodes, this was not considered to be a major issue of Pennsylvania and other northeastern states, since it was believed that our soil types were less conducive than those of southeastern states that tend to have more of a sandier soil, versus our clay loam that may limit the mobility of these nematodes within the soil and mask some of the water and nutrient issues we tend to see with plant parasitic nematodes. Additionally, there is an overall lack of awareness and testing of soil in our state for plant parasitic nematodes. And since the pressure tends to build slowly over time, it's hard to see the incremental yield losses season to season. Whereas when we start to compare this season to, say, 5 years ago, that's when the issue becomes more apparent. Additionally, climate change, as well as the use of these structures that is warming our soils, has been increasing nematode spatial range, such as the appearance of our tropical root knot nematode species, Melorgine javanica, and our southern root knot nematode species becoming more apparent and prevalent within our state of Pennsylvania. To make matters worse, plant parasitic nematodes create entry wounds in the roots through their feeding, and this can really exacerbate the symptoms of other diseases, particularly fungal pathogens that use this as an entry site. In addition to the entry wound for other pathogens, dagger nematodes are known to be vectors for viral pathogens, which I will discuss later on within this presentation. Although we did not look at this within Pennsylvania, other researchers have cited that Fusarium species, Pythium species, Sclerotinia species are all of concern within high tunnel tomato production. And though it is typically seen southern of Pennsylvania, Ralstonia solanarium can also be a huge issue within high tunnel production. I also wanted to add that ASD has been found effective for all 4 of these pathogens. I've listed here a meta-analysis of the ASD studies that were conducted at that time, although for Sclerotinia species, it may be slightly less effective due to their hardened structures. And with that, I'm going to hand over controls to Dr. DeGioia.
Thank you, Caterina. So after we have seen like what is happening in Aitana, what are some of the main soil-borne pests and pathogens that we have, the question that we have is like, how can we manage those? How can we handle them? And we have a series of options, but some of those, like Caterina mentioned, are not really practical. More and more we see that consumers and even growers are not interested in synthetic chemistry approaches like chemical fumigation. Also, like soil steaming could be an option, but is an option that doesn't consider properly like soil health, the aspect of soil health. And among all the different options that we have here, we have been focusing on anaerobic soil disinfestation. And I would like to take this opportunity to explain first how we apply anaerobic soil disinfestation, and then why we are working with this method, how this method is different from other methods. So ASD is applied in 3 steps usually. The first step is to amend the soil with a readily decomposable organic material, and ideally the carbon-nitrogen ratio of that material should be between like close to 20 to 1, 20 parts of carbon and 1 of nitrogen. And the idea is that this amendment, this carbon that we apply, is literally functioning as a sugar that will activate the soil, the soil the microbes, the bacteria in the soil, and they will start feeding on that, and then kind of consume all the oxygen and develop anaerobic condition temporarily, which generate a bunch of volatile organic compounds that are part of the active ingredient that we have. The second step will be to mulch the soil with an impermeable film, a gas impermeable film, and the scope of the film is to maintain the condition, the anaerobic condition during the treatment, at least for 2 to 4 weeks. And we can do two different type of application. We could do raised bed application, or we could do flat application, broadcast application, where we cover the entire soil surface. So what you see here is what we could do as a raised bed application. And then the third step is to irrigate the soil to saturation, which doesn't mean to flood the soil with water, but to provide water via drip irrigation for about 4, 5, 6 hours, depending on the type of soil that we have and the initial moisture level in the soil. And this helps to develop the anaerobic conditions and also facilitate the diffusion of all the organic acids and the compounds that are formed during the ASD process to go to diffuse like throughout the root zone. Now, asking the question why anaerobic soil disinfestation versus something else. I would like to discuss some of the advantages and also some of the disadvantages of ASD. ASD is a technique that is essentially an organic amendment-based approach. And as one of the advantages, and you could read more about this in this review manuscript that we published in 2020. The nice thing about ASD, besides being biological, is that it has a broad spectrum efficacy against fungal and bacterial pathogen, plant parasitic nematodes, and weeds. And sometimes we see that these issues kind of cooperate together and create more issue than the single pathogen or the single pest. And so for us, this is very important, and it almost like relate to the effect that you will have with a soil fumigant, with a chemical soil fumigant that typically has broad spectrum activity. Now, being an organic amendment-based approach, it allows us to use carbon sources, organic material that we may have available on the farm or that we may have available in our state, in our region. So here is just the example of one of the main carbon source that we are using, which is wheat middlings. And here you see the rates is about like 14,000 pounds per acre of wheat midds. And that pretty much correspond to 5,000, 6,000 pounds per hectare per acre of total carbon. Now, over the past few years, we have been working on a lot of different alternative carbon sources, and there's a lot that you could use locally, from fresh residues from the processing of vegetable to, let's say, spent grain from the brewery, or other type of like grape pomace, tomato pomace. There's a bunch of organic organic material that are byproducts of other agricultural processes, and that could be used for ASD. We also have been working on integrating ASD with other approaches, like the use of cover crops or the use of grafted plants. ASD allows us to do that, and here's the QR code for another study that we did a few years ago. And when we talk about integrating ASD with cover crops, it's important to kind of define where to place ASD before a crop, and what type of cover crop we're going to use. So this is just a scheme. We talked about the use of cover crops for ASD in a previous webinar, so I invite you to check that. The other advantage that we have with ASD is that applying this organic amendment, we are also applying some nutrients. And one of the main things that is important to know is that ASD affects the nutrient dynamics in the soil. And initially, during the ASD period, we have limited availability of nitrate nitrogen, but then as the soil goes back to aerobic condition, we see the availability of nitrogen increasing. as long as the carbon-nitrogen ratio of the amendment that we used is balanced. If we have too much carbon and very little nitrogen, then there is a risk that that nitrogen will be tied up and will not be available for the crop. So these are things that are important, but one thing that I want to mention is that in our studies, we have been able to grow tomatoes in our tunnel for an entire season without providing any fertilizer, just the nutrients that were provided through the carbon sources, and they were, released over time. The other thing that is important is to mention is the fact that really ASD is an approach that is respectful of soil health and actually promotes soil health, and that is because part of the organic amendment that we apply is not decomposed and is increasing the organic matter content and is going to be available for a series of beneficial microbes. The other aspect is that we demonstrate this in one of our recent study. We show that we have a shift of the microbial community, the soil microbial community, but then it tends to go back after having done like a cleaning of the soilborne pathogen. So that's also very important because it's like we are not leaving like the soil like as a void that then is occupied, could be occupied by other, again, by soilborne pathogens. So that is an important aspect there. The other important aspect is that compared to other methods like soil solarization or biosolarization, ASD is not as temperature dependent, meaning that it requires relatively low minimum temperature to work. Our recommendation is to apply ASD with temperature like that are relatively high, but we can see that ASD could be effective even at temperature like 15 Celsius or 60 Fahrenheit degrees. And so that allows us to apply it also in region like in the Northeast of the US. And this is an important aspect for us. Now, in terms of disadvantages, probably there is, as many advantages and disadvantages. One thing that I could mention is that, being this a biological approach, it is complex. And what that means is that often the environmental factors that we cannot control may affect the efficacy of the treatment. So the temperature, for instance, may vary during the treatment, or the soil conditions, the soil type. Those are things that, may affect the efficacy of the treatment. And knowing, the situation we are in, the type of soil, what is the moisture content in the soil, knowing what the temperature is going to be during the treatment, like planning the treatment properly will minimize any of those issues like the complexity and the potential not effective application. Another aspect to consider is the cost of the application, and that really varies depending on the organic amendment that you're using, how you implement the treatment. There's a lot of things, and hopefully we will have a dedicated webinar on just on the economic analysis of the ASD. We mentioned about the fact that we need a minimum temperature of 1560 Celsius. So like there are time of the year in which we cannot really apply effectively ASD. So the timing of the application is like you cannot always do it throughout the year. It requires some equipment. I will say is minimum equipment just to incorporate the organic amendment. But that's something that for some people could be an obstacle. And then the other aspect is that if we don't balance correctly the carbon-nitrogen ratio, we may have, let's say, we can generate nitrous oxide, which is a greenhouse gas. During the ASD process, we may have also CO2 and methane emission, but those are less of an issue. So here there's another QR code for another presentation, another paper that we published where we discuss all those things. Now, going to what we have done, so the other aspect of this presentation is like we want to show how we have done on-farm application, on-farm ASD application. And here is like a list of on-farm studies that we did in 2023. So this was the previous season. And You can see that we worked in iTunnel with tomatoes and with strawberry. We worked also in open field, and the timing of the application was, varying between like the summer and the fall. And pretty much we use always like wheat middlings as a carbon source. Here you see the rate, and we did both raised bed application and broadcast application. And some of these farm had specific soil-borne pest pathogen issue. Others didn't have clear, like, a dominant issue. In 2024, we did 3 on-farm trials, and those were one in a raspberry Aitano that then was cultivated also with cucumber. Another one was an iTunnel tomato monoculture in conventional system, and like there were 2 actually in iTunnel done with tomato production. And the application were done between the summer and the fall, and like early fall, I will say. And we used either wheat middlings by itself or wheat middlings with molasses when we knew that we had like a big issue to solve. We kind of increased the rate of like 50%, adding molasses to the mix. And Caterina is going to talk more about this, but in one case the issue was dagger nematodes, which transmit viruses. Then in the other case was verticillium and fruit knot nematode in the third case. So how we did apply the treatment, you can see here the status of the crop, the cucumber crop before we applied the treatment. This is when we did soil sampling for the dagger nematodes and also for other parameter. This is the day of the application. So the application done July 18, 2024 with wheat middlings. We use bagged wheat middlings, but normally it would be cheaper to buy the tote and distribute it, in different ways. But for us, it was convenient to do this way. So we divide, the iTunnel section and try to apply evenly the wheat middling. So in this case, this is a research activity. So we have a bunch of people helping there, including the growers and the grower and his kids. And so then we incorporate, this was an Amish grower, so they didn't want us to use any equipment that was like, different from what they normally use. So you see here how the wheat middlings is being incorporated. After incorporating everything, we set up the irrigation system, putting the line like a foot apart. And after that, we left a little section here that was kind of our untreated control right at the front. And then we used clear and black plastic. So these were the 3 different treatment that we have here. And of course, this is like, it's not research, but it's like on-farm demonstration. So we don't have replicates there, but we do repeated sampling. Now, this was the outcome after 4 weeks, 28 days. We went back to the farm, and you can see like we are retrieving all the sensors that we had installed to measure what was happening to the soil. And what you see here is that there's a bunch of weeds in the area untreated, and instead the area that was treated, there is no weeds, and that's visible also here, all the sides. Of course, there were weeds. And when we took off the plastic, this is the situation underneath. So it's pretty like the weeds are not there. And weeds are important because, especially in this case, the dagger nematodes take the viruses from the weeds and then transmit it to the plant. So managing the weeds and nematodes at the same time is an important thing. Here you see the results of the temperature. This is mainly to compare clear film and black film. And you see that with the clear film, it was like the temperature was a little bit higher. We reached, over like 50 Celsius degree. But on average, it was around 40 Celsius degree. And this is a graph showing how anaerobic the soil was over time. So we start that the soil is aerobic above this 200 line. and then it goes down immediately within few hours, and we reached very good anaerobicity level. And between the two film, there was not much of a difference, except here when there was an event for which the plastic was lifted, and that was because of rain outside. And uh, in this graph, you can see what is the cumulative redox potential, which is a measure of overall during the 4 weeks, the 28 days, how anaerobic the soil went. And what you can see here is that in this case, the black TIF was slightly higher than the clear TIF, but that was really due to this difference here. If we don't take account of this period, essentially the two type of plastic were generating the same level of anaerobicity. And the control in this case went also anaerobic for some time. And the reason there was because we applied water. The sensors were also in the area where we applied irrigation. So that was the effect of the water itself. Second farm study, pretty much similar situation. We applied in this case with middlings and molasses combined. This was the farm that had the verticillium issue. And the grower got really creative about how to apply the molasses. He mixed it with water in a tank and in a spray tank. And then he had built, after we talked about it, a system to distribute the molasses throughout the plot. You can see he created like a PVC pipe, drilled some holes here, and he could have drilled more, and that would have been faster. What we ended up doing was to apply half of the molasses in this way, and then the other half we applied it through fertigation. Now, keep in mind this is molasses mixed with diluted with water, at least 1 to 1. Here you see the black tarp and the clear tarp covering the two section of the eye tunnel, and also in this case we had a control. Here we see also some slight higher temperature in the clear film, but not really a big difference. And this is again the anaerobic the soil redox potential, which is a measure of how anaerobic the soil went. And you can see that it went pretty anaerobic, and there was not much difference between clear and black plastic. Also, in this case, the control went anaerobic just a little bit, but then it stayed pretty much aerobic, so in presence of oxygen. And here you see the effect. Clear film in this case was a little bit more anaerobic than the black TIF, the black film, over the entire period. Third case study, it was the grower that had the nematode issue, and Caterina is going to talk more about this. But the main thing I would like to mention here is that when we did the pre-ASD soil sampling, we realized that in this case the numbers of nematodes were really high, and so we decided to go again with a combination of wheat middlings and molasses together. And so we applied like 1.5 the rate that we normally would apply of carbon. And so you see here half tote and the bags of wheat middlings that we distributed. This was at 32 by 200 feet eye tunnel, so it's like double the size of most eye tunnels. And you can see here same picture, like distributing all the wheat middlings evenly. And we did that by hand. Then everything is incorporated in the soil with some machinery. And then we applied, we set up the drip irrigation. In this case, we went every 10 inches rather than every foot, just to have more coverage of the water. And all the molasses was applied after dilution with water. It was applied through the drip. So this took a little bit longer to apply the molasses, but it was done while we were irrigating. So at the same time, you are irrigating and applying the molasses. And this is how the field looks. Of course, we had some sensors back here, and this is the graph showing the temperature of the soil. And again, the clear film was slightly higher, but not much of a difference. And you can notice that as we went from the summer to the fall, the temperature that we reach is lower. Before we were like reaching 50s. Now we are maximum at 40s, but we have also like lower temperature here toward the end of the 4-week period. Here you can see the results of the soil redox potential, and what you see here in this case is that the clear film was more anaerobic than the black film, substantially more anaerobic. And that could have been for the slight difference of temperature, but this is what we observed in this case. And so there was a clear difference between the two type of film. The clear film was much more anaerobic over time, cumulatively, compared to the black film. Now, when we retrieve the film, this is what we see. The soil turned almost like black, and that is like for the reduction of the iron and other metals that are in the soil. So it's very interesting to see how the soil looks right after you uncover it. And this was, 4 weeks later. I think here I'm gonna switch again. I'm gonna give the control to Caterina, and she will show us the results that we got from those on-farm studies.
Okay, thank you so much, Francesco. So starting off, I'll be discussing the site that Francesco just ended on. This is a Pennsylvania high tunnel that has been continuously cropped with tomatoes for more than 5 years. And it was also included within the Gugino Lab soil survey that I discussed at the start of this presentation, where even back in 2021, this high tunnel had high levels of root knot nematodes above that economic threshold, as well as having a positive detection of Pseudoperennichita terrestris, which is that causal agent of corky root rot. And again, just to reiterate, this was whole surface treated. That's mainly due to the nematodes being mobile in the soil and really wanting complete coverage of the treatment so that we're not leaving the areas in between the rows as potential inoculum sources. Again, this was treated with 1.5 times the standard rate to really get that anaerobicity as long as possible, as low as possible, with the half the tunnel being covered with black plastic and half being covered with clear. As far as my data collection was conducted, we collected soil samples both before and after the ASD process, and the high tunnel was divided into 24 blocks of approximately 7.5 by 32 feet in size. And then within each block, composite soil samples of about 10 to 20 soil cores were collected using this zigzag pattern. This is really used to minimize the potential spatial bias that we see with soil-borne pathogens, where there tends to be hot pockets within the field that have more severe symptoms versus other areas having lesser symptoms or no symptoms whatsoever. So this really gives us an indicative picture of the entire surface. And then once that soil sample was brought back to the lab, it was used for 3 separate analyses. The first being used for bioassay plants, in which we directly seeded tomato moneymaker seeds into the soil and grew those plants for 8 weeks before the plant was destructively harvested and root symptoms were evaluated for root rotting and galling. The next subsample of the soil was used for soil nematode counts to assess the plant parasitic nematodes. And then the last subsample was used for soil DNA extraction, with downstream application being diagnostic testing using PCR for the Colototricum cucoides and Pseudoperennichita terrestris. Getting a little bit more into our bioassay plants, I rated these plants on 2 separate scales. The first being Zeke's galling scale for root knot nematode galling, with 0 being the roots were entirely clean and had no galls, and 10 being that there was no clean roots left and the entire root system was galled. Next, I used a scale designed by Dr. Anna Teston's lab at Ohio State University. This is a tap root rot rating scale in which 1 is a healthy taproot displaying no lesions, and 5 is that the taproot is either completely missing or rotted. Additionally, I recorded the percentage of fibrous roots that were rotted and extracted nematode eggs, as well as counting them under the microscope. And so first and foremost, looking at the number of J2 root knot nematodes within the soil, like Dr. DeJoya touched on, pre-ASD plants averaged about 2,120 nematodes per 100 cc's of soil. And to give an idea about how severe this is, it's over 200 times what we consider to be the actionable threshold for root knot nematodes. So this is a really severe instance that we wouldn't typically see within the field. Post-ASD soils dropped, post-ASD root knot nematode counts dropped by over 50% within the soil, with post-ASD soils averaging 996 J2s. But it is important to note that all of the J2s present post-ASD were dead, and there was no significant difference between plastic types post-ASD. To give a better picture about how these dead nematodes looked, on the right here we have what would be a healthy root knot nematode, in which it is typically moving under the scope, and you can see that the cuticle is smooth and the organelles can easily be distinguished. On the left here is what Dr. Cantor saw post-ASD with these root knot nematodes, where they take this C pattern when they're dead, as well as the cuticle being distorted and the organelles not being readily distinguishable, almost looking like this nematode had been boiled. Additionally, he noted that on a good portion of the nematodes, there were what appeared to be bacterial colonies growing from the nematodes. However, it's impossible to say if this was a secondary infection that occurred post-mortem, or if this may be one of the mechanisms that ASD was decreasing that population. Moving on to the bioassay plants from this site. On the right here, I have what is typical of the bioassay plants at that 8-week time point. And I wanted to share this as a comparison to the pre-ASD plants, in which you can see severe plant stunting, both of the root system and the above ground plant. These plants average 6.7 of a galling index, meaning that over half the plants were galled with root knot nematode galls, and 8 of the 24 plants died before harvest, likely due to the severity of these root symptoms that we're seeing. Post-ASD plants were much healthier in appearance, with black plastic treated plants having a galling index averaging 2.4 and 2 of the 12 plants having no galls whatsoever. Whereas the post-ASD treated with clear plastic had a galling index of 0.8 and half of the plants had no galling whatsoever. So there was a significant difference between the plastic types, with lower levels of galling within the clear plastic. And this is kind of some pictures showing what the pre-ASD plants looked like on the left, their root systems, compared to the post-ASD plants on the right. And there was still some minor galling, but substantially more vigorous of root systems and overall much healthier. Looking at the number of root knot nematode eggs, this is important since this is a measure of the nematode reproduction. All of the pre-ASD plants produced had extracted eggs, averaging 978 eggs per root system, whereas only 1/3 of the black plastic bioassay plants contained eggs, and 1 of the 12 bioassay plants contained eggs. It is important to note that within this variable, the plastic types were not statistically different. However, they were statistically different pre versus post treatment. Next, moving on to our taproot and fibrous root rot ratings for this site. The taproot rot was significantly reduced by the ASD treatment, with pre-ASD plants having an average taproot rot of 4.4, which again, 5 being that taproot is either completely rotted or missing, and post-ASD plants averaging 1.5. So that's close to that 1, which is healthy. Additionally, the percentage of fibrous roots that were rotted was also significantly reduced, with pre-ASD plants having an average of 50% of their roots rotted, and post-ASD plants having an average of 16.7. And I've highlighted here in the red box what some of that fibrous root rotting looks like with the lesions. But again, within this variable, there was not a significant difference between plastic types. Then looking at my molecular detection of Colototricum cucoides and Pseudoperennichida terrestris, Colototricum cucoides decreased post-ASD by 73% within black plastic treated blocks and 81% within clear plastic treated blocks. Additionally, the detection of Pseudoperennichida terrestris decreased post-ASD by 75% within the black plastic treated blocks and 100% in the clear. And so switching gears a little bit to go to that site JA1. This site was the site that Dr. DeGioia talked about that was growing the raspberries facing viral problems. He pulled these raspberry plants and planted cucumbers, but was still having issues. And again, it was a whole bed treatment with the aim of really managing those nematodes. And the dagger nematodes are particularly an issue on our perennial fruit crops as they significantly reduce both the yield and fruit quality, often rendering our fruit crops useless over years as this issue exacerbates. And so pre-ASD blocks had an average of 27 dagger nematodes per 100 cc's of soil. And although this sounds like a small amount in comparison to what I discussed with the root knot nematodes, it's really important to mention that that economic threshold is much lower because of the viral transmission that they're causing. Post-ASD blocks average 0 post-ASD, with only one block containing a single dagger nematode. So a substantial difference than what we were seeing post-ASD. And looking at our bioassay plant symptoms, within this site, the root systems were overall quite healthy pre-ASD. So for the sake of time, I didn't share those values. It was really the viral issue that was the problem within the site. And so 3 out of the 20 pre-ASD bioassay plants from this site produced viral symptoms. The picture to the right here is some of the viral symptoms that were appearing on my tomato plants with very characteristic ring spotting. I then performed ELISA immunoassay strip detection for tomato ring spot virus on these plants, which came back positive. However, when we compare this to the post-ASD plants, 0 out of the 20 plants had viral symptoms. And again, I tested a subset of those plants to ensure that they weren't positive but asymptomatic. And all of the post-ASD plants tested came back negative. This site did not contain Pseudoperennichida terrestris, but within the Colototricum cucoides, detection decreased by 92.3%. And for this grower in particular, because he would like to plant raspberries again, we would like to come back in prior to planting and perform additional nematode testing to make sure that that dagger issue has stayed low to non-existent in prevalence. And so as a discussion and conclusion, as well as future work for the on-farm soil-borne pathogen piece, ASD was found effective to reduce the detection of Colototricum cucoides, Pseudoperennichida terrestris, dagger nematodes, and root knot nematodes, as well as reducing the root rot and root knot nematode galling within site LN2, which was significantly lower post-ASD. Additionally, the number of root knot nematode eggs extracted from the bioassay plants also significantly decreased. And particularly within site LN2, the clear plastic performs somewhat better, but as Dr. DeGioia mentioned, that's likely due to the higher amount of cumulative redox potential within the clear versus the black. We would like to visit both sites after this year's upcoming crops to discuss with growers how they felt the overall efficacy of ASD was, as well as to look at their crops and make sure that the management of these pathogens remained as we're seeing within these this data. And lastly, we'd like to continue to conduct more on-farm ASD trials, since this not only increases the data available, but also increases grower knowledge, which is the main goal to potentially increasing adoption for the growers. And here's some of the cited works that I used within this slide. And I'd like to really thank and acknowledge my advisor, Dr. Beth Gugino, as well as my committee members, Dr. Francesco De Gioia, for really being the lead of all of our on-farm research, as well as Dr. Mihail Cantor and Dr. Sharifa Crandall, as well as our funding sources, USDA NIFA in particular. And again, if you haven't already, please fill out the survey for soil health if you're a grower that is interested in the ASD process. And with that, we thank you for listening and would be happy to answer any questions.
Okay, great. Thank you very much, Caterina and Francesco. And we are going to move on to questions. Based on your experiences, can you tell me about the even distribution and volume of the water used?
We talk about the equivalent of 2 inches of water, and that would be 5 centimeter, the equivalent of 5 centimeter of water. And so we, depending on the drip tape that you use, the flow rate that it has, you can estimate like how much, like the total volume that you need to apply, and typically is between 5-6 hour maximum of water. Sometimes even 4 if the soil is already wet. So that's pretty much when we are doing raised bed application, we like to put like chew drip tapes underneath the plastic, the raised bed, because that will allow a better coverage. And when we did the broadcast application, we saw sometime that like 1 foot, like putting the drip tape 1 foot apart allows to have good distribution of the water, but because there's like up and down, the soil is not completely even after sometimes is the carbon source is incorporated. To have really like to make sure that the water is distributed evenly, we suggest like 10 inches to put the drip tape 10 inches apart, which is a little bit more, like a few more lines of drip tape, so that doesn't cost much. So it is important to add the water because all these like the anaerobic conditions really benefit from the development of anaerobic condition, really benefits from having the soil wet, and also the microbes, they need water to do what they are doing. And again, like I said at the beginning, the water is also a way to have kind of the compounds that are generated, the active ingredient that are generated, distributed, like to distribute them throughout the profile of the soil.
Okay, great. Okay, here's a question about the sensors. How deep were were the sensors that were measuring the redox potential?
Yeah, the sensors that measure the redox potentially typically it's like I believe it's like 4-5 inches. Like yeah, it's like we're talking about 12 centimeters, something like that. It's just the length of the sensor. You don't need to push it further. It's just the length of the sensors. Like our interest is to be within the root zone. Uh, for the temperature sensors, we're talking about like 5 centimeter, a couple of inches.
Okay, great. Wow, we've got a lot of questions coming in. That's wonderful. Okay, so in succession plantings in a high tunnel, what is the minimum number of weeks you would need to wait before replanting if you're doing something like the molasses and conventional tomatoes study?
Yeah, that's a good question. I I'm taking those questions, but Caterina, feel free to jump in if you have things to add. Um, I think, in general, we try to apply the ASD in 3-4 weeks. If you are working with AV soil, it may tend to be more like 4 weeks rather than three. If you are in a sandy soil, lighter soil, it could be 3 weeks very easily. What is important there is like if you need to terminate the treatment in 3 weeks, what is important is that you aerate the soil. So removing the plastic, if you do a broadcast application, will stop the treatment. So the soil will go back to aerobic condition and you can plant again your crop. When we work with raised bed, the plastic, we don't remove the plastic. We plant into the raised bed. And what is important in that case is to punch the holes, the planting holes, and let the soil breathe for a couple of days. As long as, the soil is still like, it's not saturated with water, but it's like just, normally wet, you should not have a problem there if you aerate it for a couple of days. So that is, an important aspect is you don't want to plant if the soil is still anaerobic. And you when you open the plastic, when you remove the plastic, if the treatment was effective, and sometimes even without removing plastic, you will see that it smells a lot. And that is all these like volatile organic compounds that are being generated. Um, but once you let the soil breathe, it goes immediately to aerobic conditions again, and in a couple of days you can plant.
Okay, let's see. Can the plastic be reused each year for ASD?
Yes, so when we use the raised bed system, that plastic is used like a regular mulch, so you will not be able to reuse that more than a couple of cycles. We do double cropping in that case. When we are talking about the broadcast application, we use silage tarp. Those are either like the black one is typically 6 mil. The clear one that we found that is also impermeable to oxygen is like 2 mil. The 6 mil, you could use it over and over, no problem. The only issue there, you need a place to store it, and it's gonna be a place like the plastic is gonna smell because of the treatment. So you need a place that it's gonna be ventilated outside for some time, and then maybe you'll be able to bring it in. But yeah, definitely the advantage of using the tarp is that you could use it over and over. The clear plastic, it's cheaper than the black one because it's like a thinner plastic sheet, and it could be subject more to, like it could be broken maybe, but like it doesn't really tear. It's easy to take it, without tearing it, to take it out of the soil, and it could be reused at least, I would say, a couple of times. Um, One thing that is important, because I know of some growers that attempted to do this on their own, make sure when you do the treatment to seal the sheet all around it. And you could do that manually with some shovel, or you could do that also with some equipment. There's like some attachment to BCS tractor that you could put there to dig the furrow and then cover again the sheet and the plastic.
Okay, yeah, we had a question about exactly that. It says, how thorough does the sealing of the plastic have to be? Is it sensitive to leaks on the side or small holes in the plastic?
Yeah, we try to reduce any leak. We try to cover the side really well. If we, let's say, accidentally punch the plastic, then we will tape it. Like for instance, to put the sensor, we go through the plastic and then we tape it right after, and that's enough. Overall, like if there are tiny leaks, it's not a problem. But if you don't close the side of the plastic, that's not going to be helpful. It will not achieve the level of anaerobicity that we need.
Okay, yeah, we have some specific questions about that type of thing. So, Dave here was asking whether it would work if the plastic was draped over heating pipes are 7 inches high in the pathways. Yeah, over every path between the beds, and what would be the impact if you only covered the soil for 2 weeks? So, I guess the first question was, does the plastic have to be touching the soil, or can it also be? Um,
Yeah, the
yeah.
the more adherent to the soil is, the more like, it's like there's air gap the better it is because we are trying to generate anaerobic conditions. Um, that is easier to do with sandy soil. Aviar soil will tend to form like big clumps, and that may, leave the soil a little bit like of space, airspace there. That typically is not a big issue, but we try to minimize that by preparing the soil, in a way that it's going to be good, easy to lay the beds.
Okay. Let's see. Yeah, and then the other question there was what would be the impact if you only covered the soil for 2 weeks?
Yeah, like I said, there is not like um, a rule on how long it should be there, and a lot of studies have shown that what is important is the level of anaerobicity that you achieve within the first few days, rather than how long it stays anaerobic. But I think that we still don't have clear data on that. If terminating the treatment within 2 weeks is a requirement because you have limited time between crops to do the treatment, it could be done. I know my colleagues in Florida, Dr. Shinzao, has been testing that solution, and they were successful doing it with leafy greens grown in high tunnel. But like I think depending on the type of pest that you are dealing with, um, a longer period may be or may not be needed. The critical thing is when you're working in a heavy soil, if you exceed with the water amount that you should apply, then you know the fact that the soil stay wet could be a problem in returning with a crop after 2 weeks. But again, the solution there is to aerate the soil and then remove the plastic, and then you can work the soil again, you can plant your crop.
Okay, if you do only the beds but not the paths, do the problems move back in quickly?
Yeah, so it really depends on what the pathogen is. In the case of root knot nematodes and other parasitic nematodes, the problem might move back in quickly. But something like a fungal pathogen, it might not be as quick. I think it is important to note your tillage practice too. I mean, if you're tilling the entire bed, the entire surface of that tunnel, and you're not treating in between rows, there's always the potential that you're mixing in that inoculum. So I think it really depends on the issue at hand, the overall pressure. Yeah, those are kind of the two main constraints. I'm not sure if you want to add anything else, Francesco.
Yeah, no, I think that what you said is, that's the answer. It really depends on the pathogen. So root knot nematode may be more challenging because they move like a lot vertically and spatially. And so, talking with the growers, we see more of a benefit in that case to do a broadcast application. In other situation where, also like if the time is limited, we want to do the application in relatively short time, then it may be more convenient to have the raised bed so that you have already the plastic where you're going to grow your crop. And so those are the things to consider, in terms of advantage and disadvantage.
Okay, Katerina, you had started typing an answer to the methodological question about nematodes, how the pre and post levels were measured. Do
Yeah,
you want to talk about that?
I just finished that answer, but
Oh,
I
okay.
couldn't say out loud what I said. I said
Okay.
this is a great question and a potential limitation. Our growers are always asked to remove the vegetative material prior to us coming, and we perform the soil sampling the day of the ASD process application, as well as the day of plastic removal post ASD when the soil is bare roughly 1 month later. Additionally, we don't compare between sites partly due to the timing difference. And another component to the on-farm trials in general is just that grower knowledge is a huge factor in this. And a grower is really the expert of their site. So they're good at telling us how this subsequent crop compared to prior crops. although this isn't something we can quantify. I hope that helps.
Okay, great. Okay, so here's a question about doesn't the organic matter contain pathogens? And in anaerobic conditions, can't that favor the survival of some pathogens? So is the organic matter sterilized before application?
I could say that, yeah, no, the organic matter per se doesn't contain pathogen. I mean, it could contain some pathogens, but like we are working with residues and stuff that are not like may not carry the same type of inoculum that affects. Like if you're working with wheat middlings as a carbon source, it doesn't have anything to do with the pathogens that affect tomatoes.
Mhm.
But that's typically not a problem. And anyway, like um, there is only limited cases in which there's certain pathogens that could take advantage of some specific carbon sources. If you don't achieve good anaerobicity level, they could not be affected or slightly promoted um, by. But in that case, again, it's like um, if you don't apply it correctly, if you don't achieve anaerobic condition. A lot of the soil-borne pathogens have developed to coexist with the plants, and the plants need oxygen. And so most of them will not survive if you reach good anaerobic conditions.
Okay, so here's a comment, great information, and a question. With concerns about microplastics and polyculture, have you done or do you plan to conduct ASD with biodegradable plastic mulch, and do you think that would be feasible? I mean, right now there's no biodegradable plastic mulches which are permitted in organic production, so that probably wouldn't be part of this project, but maybe a different project.
Yeah, I would say that is something that we have considered, and we have been talking with company that manufacture biodegradable mulch to see if there is anything that we could use that will allow us to, let's say, do the treatment and then decompose. But so far, we have not found like material that really is suitable for these. There's no total impermeable biodegradable plastic. There's regular like biodegradable film. And I know that Dr. Erin Roscoff, who is part of our team, and she's a researcher at USDA in Florida, has done some testing on that. And you know, that would be a question for her and what she found out and what they found out. Um, yeah, and but it's something that we have considered, but I think the technology is not there yet for us to use it.
Okay, great. How deep do you need to incorporate the carbon source, or do you just put it on top?
Yeah, typically our interest is to treat the root zone. And so, for tomatoes, that could be the top, 20 centimeter.
Let's see. Going back to I guess the first question about irrigation, could drip or sprinkler application and soil tillage make a difference in nematode control? What do you think about that?
Yeah, I mean, our objective there is to really like distribute the water evenly. With the sprinkler, possibly you could do that. The issue is so what we tend to do is to put the drip underneath the plastic when the soil is still relatively dry, is dry. We start running the water before we put the plastic, but you can still walk around and then we try to seal everything while the soil is still dry. If you were to use sprinkler, the soil will be wet, and then to cover the plastic with that soil, it may be challenging.
Hmm.
So that's why we prefer to use the drip. In other situations, like in Florida, for instance, there is land where you could use sub irrigation. That will be another method, like where you raise the table of the water, and you have a sub irrigation. Essentially, the water is coming from the bottom of the soil.
Okay, great. Let's see. We have a question from Timothy who's wondering how pre-plant nutrient application might affect ASD performance.
Yeah, that's a good question. So typically, what we try to do is uh, the moment of the application, growers we don't apply any other fertilizer. We just go with we try to balance the nutrients with the organic amendment that we are using. So, for instance, wheat middlings is like a carbon source that has a perfect, CN ratio. It's like 122 to 1, something like that, or 20 to 1. So it's perfect. You don't need to balance the nitrogen there. When we work with molasses, it may be beneficial to combine that with chicken manure to balance the carbon nitrogen ratio, which is going to guarantee that after the treatment we have a good availability of the nitrogen. Now, when a grower is planting the crop right after, and typically they do like fertilization pre-planting, we kind of give them like an estimate of what we are applying in terms of nutrients with the organic amendment, and we tell them that we don't recommend adding more nutrients. But some growers will still want to do what they want to do, and so in those cases we try to have, let's say, to have the grower control versus our treatment, so that they can see um, how nutrient wise the impact of ASD on nutrient availability.
Great. Okay. Could it benefit ASD to irrigate more than the initial window and molasses plus molasses during the 4-week window to increase anaerobic conditions?
Okay, so if I understand correctly, the question is like, if we add molasses and more water during the treatment, will that continue to increase the anaerobicity level? Well, the answer is yes, but typically that is not needed. It's enough for us to do, the treatment once, apply the molasses or the carbon source once, and the water once. Now, if you keep adding water, the soil will just stay more anaerobic. If you keep adding carbon source like molasses via drip, you are increasing potentially also the anaerobicity, but then you are also prolonging the treatment. And that's typically not what a grower wants. Now, there are circumstances in which, like, let's say we apply the ASD treatment in the fall, and the grower is planting tomatoes again, like in the new year in the winter, and so there's a lot of time there. But typically, what we recommend is that after the ASD treatment, if we have done the broadcast application, they plant a cover crop, so that that the soil health will benefit also from the cover crop.
Great. Let's see. Can ASD provide long-term suppression, or is it only effective for a limited period?
Yeah, that's a good question too. I would say like we need to compare ASD with other treatments. And for instance, we know from the literature, from different experiences, and also talking with growers, that if you do soil steaming, you will need to steam the soil every year. And that is because you are killing all the beneficial and the bad stuff. But again, you are um, destroying the balance that, it's part of the soil health. And, that soil may be occupied again by the pathogen very easily. And so you have a problem there. When you do fumigation with chemical fumigant, it's pretty much the same thing. The soil is like people say is addicted to chemical fumigation. And that's the reason there. If you don't do the fumigation every year, you will not see the benefit after one season. In the case of ASD, I believe we see the effect at least for a couple of seasons. I had growers telling me after 2 years that they saw the effect of ASD, after 2 years. We don't know, what happens if we go over the 2 year period, but the way I explain this is, again, one important aspect of a anaerobic soil disinfestation is that we are shifting the soil microbial community, meaning that you have different groups that prevail at different moments during the treatment, but you never kill everything. You never create a void. And so you are cleaning like the bad pathogen, the what you want to suppress, but still there is, some type of microbes there that are resisting to the anaerobic conditions, and they are living also afterwards. They are thriving because we have applied organic amendment, and that organic amendment is really helping, the nutrition of those microbes. And so I will say that that's the important difference there. And typically, we like, for instance, I probably am gonna go to visit the grower of the nematode farm that we showed here tomorrow morning, and I was talking with him this morning on the phone, and the plants are doing well and everything. Like typically, he's been able to, because we have done more application with him. He's been able to work with soil after ASD for several years without having to go back with ASD, and that is a good thing.
Great. Okay. Looks like we have one more question here in the queue. What are the temperatures achieved and how deep do they go?
Yeah, I mean, we typically measure between 5 and 10 centimeter. In the past, we used to put a lot more sensors because we were like interested in, investigating that. I would say, deeper you go, lower will be the temperature. But over the summer, it's like we can exceed 50 Celsius degree, which is a lot. Consider that is temperature, and I'm talking about both the clear and the black film. Typically, that will be a temperature that will be considered like soil solarization. And so we measure that around 5, 6, 7 centimeter. If you go deeper, I expect that is not going to be as high, but I expect that it's going to be around 40, for instance. And deeper you go, it will be lower. Uh, in all these, like the water component, like the irrigation water that we put there, I mentioned before that we want to treat like the top 20 centimeter, like the root zone. But when we irrigate, we may go a little bit deeper than just where we incorporate the the carbon source. And if you use like a carbon source that is liquid, like molasses or ethanol, which they use in Japan, that may easily reach the 30 centimeter. So, and because we are not concerned, so the main objective of the ASD treatment is to achieve anaerobic conditions. And it's not the control of the pathogens is affected by the temperature. Higher is the temperature, more we can cook nematodes, more we can control pathogens. But even if we were at 30 Celsius degree, that would be fine. 25 Celsius degree will be fine. The treatment will still be effective. If we go below 20, then the efficacy may be limited.
Okay, yeah, we got a last question about the optimal temperature. Is there an optimal temperature for treatment?
It depends on the pathogen. So there are pathogens that will be controlled at relatively low temperature, and others, like if you're dealing with something like Phytophthora, my colleagues that are plant pathologists that have worked with these tell me that you need to achieve high temperature.
Okay, great. Thank you, everyone, for all these really great questions. And as I mentioned, the recording will be available within a week on the eOrganic YouTube channel, as well as on the project website, which is linked to the eOrganic website. You can go to eorganic.info/ASDeasy if you can remember that. But otherwise, if you just go and you Google ASD Easy Organic, you should be able to find the project website. And we have the other webinars about that are part of the series on there, and we'll be doing more before the project is over. So we'll look forward to that, and we'll let you know about those. So thank you so much, Francesco and Caterina, for sharing your research with us today, and thanks to everyone online for coming.
Thank you. Thank you, everyone.
Thank you.